We're very excited to have with us Dr. Eric Chang. He's going to be speaking about the vagus nerve and bioelectronic medicine. Dr. Chang is an associate professor at the Feinstein Institutes for Medical Research at Northwell Health. He's a neuroscientist whose research focuses on communication between the brain and body along the neuroimmune axis. Dr. Chang's lab investigates how the nervous system, particularly the vagus nerve, encodes and transmits immune and visceral signals to the brain. And this work aims to advance the field of bioelectronic medicine, which explores using devices to modulate neural activities for therapeutic purposes. Dr. Chang earned his PhD from NYU and completed postdoctoral training at Weill Cornell Medicine's Burke Neurological Institute. He completed his undergraduate at UCLA. And I will drop his recent paper into the chat. Feel free to drop questions in the chat during the call, and he will take he'll take questions at the end as well. Thank you so much. Really appreciate you joining us. And we've been long-term fans of Feinstein's work, Northwell's work, Kevin Tracy, and we look forward to this talk.
All right. Thank you very much, Monica, for the invitation, and hello to everyone online. I'm going to tell you about some of the work we're doing here at the Feinstein Institutes in Long Island, New York, focused on the vagus nerve and this emerging field of bioelectronic medicine. So very broadly, our research focus of our lab is on interactions between the nervous system and immune system, sometimes called neuroimmunology, but that means different things to different people. Basically, it's how the nervous system and immune system communicate with one another. Now, if you had gone to med school, maybe 20 years ago, you'd learn about the nervous system in a module and then the immune system in a separate module, and basically be told they don't talk to one another. The nervous system's immune privileged and it's separated. But work really in the last 15 years or so has really shown that's not the case, that there's actually very rich interactions between the immune system and nervous system in many different compartments of the body. So you can see, both in the central nervous system and the peripheral nervous system, the central nervous system being the brain and spinal cord, periphery being everything else, at the nervous system level, you can see there's interactions between immune cells, releasing immune mediators, talking to nerves or neurons. What we focus on a lot in the lab is, particularly in the periphery, which includes a nerve called the vagus nerve, how are different immune mediators, such as cytokines, but there are other immune mediators, how do they interact with nerves? How do then nerves and neurons encode the information about the presence of cytokines, how much cytokine there is during inflammation? And what we know in different disorders, particularly chronic inflammatory disorders, obviously in autoimmune disorders, including potentially type 1 diabetes, there's a dysregulation and imbalance in this communication between the nervous system and immune system. So, the reason why we study inflammation in the body as one of the main focuses of the lab, if you look on this schematic, you can see diseases that involve inflammation in the body include basically every major disease you can imagine: cancer, cardiovascular disease, autoimmune disease, neurodegenerative disease. And then, of course, under autoimmune diseases, the major ones, not a comprehensive list, but the major ones include lupus, rheumatoid arthritis, MS, inflammatory bowel disease, and then, of course, type 1 diabetes. And if you look at the bottom of this schematic, you can see the potential contributors and then, therefore, therapeutic targets for treating inflammation in the body include targeting cytokines and chemokines, and this balance between pro-inflammatory and anti-inflammatory mediators. And so what our lab works on, and the people I work with here at Feinstein, is what is the role of the nervous system in particular, both central and peripheral nervous system, in monitoring and regulating inflammation. So those who aren't familiar with the vagus nerve, it's cranial nerve 10, one of the 12 cranial nerves in the body and in the nervous system. It's actually a paired nerve that descends from the brainstem, and you can see it's the longest cranial nerve in the body. Innervating our major visceral organs. Vagus is Latin for wandering, because you can see it doesn't go down in a straight line. It kind of goes to innervate various different organs, but wandering not in a random way, in a very directed way. It's a major part of the parasympathetic branch of the autonomic nervous system, which is thought to be the automatic nervous system, things you don't think of, such as breathing, heart rate, glucose levels, which I'll talk a little about later, many different gut functions, and autonomic reflexes, of which, again, I'll talk a little bit about. Interestingly, a majority of vagus nerve fibers across the mammalian species are actually sensory afferents. So they're glutamatergic, they send signals from the viscera and the body into the brain. So about 25 years ago, work from the Feinstein Institutes in Dr. Kevin Tracy's lab here identified a reflex circuit called the inflammatory reflex that involves the vagus nerve. And in this part here on the left, sometimes it's called the cholinergic anti-inflammatory pathway. You can see that, and the paper was a Nature paper published in 2000, so literally 25 years ago, in a rat model of endotoxemia. So in this model, they inject rats with LPS, lipopolysaccharide bacterial endotoxin, and induces inflammation acutely. And in those rats, when they stimulated with electricity on the cervical level of the rat, that reduced levels of tumor necrosis factor, basically reduced levels of cytokines, and inhibited inflammation, protected those rats from lethal endotoxemia, so improved survival. And through a series of papers from the Tracy lab and many other labs around the world, we know that this is a cholinergic circuit descending from a brainstem nucleus that I'll tell you more about, going down to the celiac ganglion complex, then ultimately affecting cells in the spleen to reduce cytokines. And just like in any reflex circuit, as we learned from Charles Sherrington, there has to be also a sensory input to the reflex that triggers it, right? When we get a pinprick on our hand or touch a flame, you withdraw. So that sensory input was kind of, while we knew a lot about the motor efferent, the output, the sensory input, specifically how does the brain and nervous system know about inflammation in the body, was one of the major focuses of some work that we did in the lab recently, and is related to the paper that I'll go over now. But as I already mentioned, the vast majority of fibers in the vagus nerve are sensory afferents that go up to the brain. So what we wanted to know is, when there's immune mediators being released by immune cells, including cytokines, how do sensory neurons and nerves detect that event, and then do they encode it in some form of neural activity? Because neural activity is the currency of the nervous system, so presumably there's some way for the nervous system to know that there's a release of cytokines and inflammation at a particular site. What we know, so the vagus nerve, as I already told you, innervates our major viscera, including the pancreas, shown here. So if there's inflammation in any of these sites, let's say pancreatitis, for example, you will get presumably a release of cytokines here. And we know that the vagal sensory neurons, so the cholinergic neurons that make up the motor efferent arm, are actually in this dorsal motor nucleus here, but the sensory afferent cell bodies are in the vagal ganglia. In the mouse, which is work I'm going to tell you about, it's a fused structure, so it's technically the jugular ganglia and nodose ganglia. For simplicity, we'll call it vagal ganglia. So that's where the cell bodies are. And work by Tomas Huerta, working in our lab while he was first as a PhD student, what we did was develop with Tomas a technique to image these vagal ganglia, focusing on actually the nodose ganglia, which is a subpopulation of that structure. And what we did was use something called a calcium imaging miniscope, or miniature microscope, which was an open source technology development from a lab at UCLA. And basically use that to image the vagal ganglia in mice that have a genetically encoded calcium indicator called GCAMP6F in the glutamatergic neurons. So this is kind of what it looks like. A mouse is under anesthesia. We perform a surgery to open up the neck around the trachea. The vagus nerve travels along the carotid artery in both mice and in humans. And so Tomas has a mouse under anesthesia. This is the miniscope. It emits blue light and collects in the green emission spectrum. So we can see, and in these experiments, what we do is put capsaicin actually on the nerve at a very low concentration on the vagus nerve. And capsaicin activates TRPV1 channels, a type of TRP channel. Capsaicin is what makes chili peppers hot. If you put that on the nerve and image at the level of these vagal ganglia, you can see many neurons light up, because many of these neurons are nociceptors. They mediate pain and things that are changing, particularly when something like hot chili pepper is or changes in temperature are happening in the body. Those are mediated by TRPV1 channels. And in this paper that we published in 2023, we developed a pipeline to allow other labs, if they're interested, to use a miniscope and use Python-based analysis to extract regions of interest, which are the neurons, and then perform different filters to see the neurons. And then More recently, in the paper that Monica mentioned that we just published earlier this year, we applied this approach to actually look at cytokine signaling. So again, we're using a miniscope, imaging the vagal ganglia, and then now we're putting on 3 different cytokines. So TNF, tumor necrosis factor, interleukin-1 beta, are pro-inflammatory cytokines. IL-10, interleukin-10, is an anti-inflammatory cytokine. And here on this video, you can see this is the nodose ganglion neurons' responses to TNF, which you can tell already that they look qualitatively different than the capsaicin videos. If we do these are actually videos from separate recordings in different mice, but if you look again at a qualitative level at the changes in fluorescence, so again, this is a GCAMP6F indicator, and each cytokine is being applied to the vagus nerve, and you can kind of see at a qualitative level that the IL-10 and TNF and IL-1 responses are slightly different from one another. You can see, for example, IL-1 beta has low changes in calcium fluorescence, but they look a little bursty. So that, maybe a lower amplitude change in fluorescence, but, a different type of response than TNF, which has kind of a larger amplitude, like you can see those. And each of these kind of things lighting up are different neurons, vagal sensory neurons in the nodose ganglion, responding directly to cytokine application on the vagus nerve. So we can quantify this in the form of calcium transients that look like this. And you can kind of see when we average them across many different applications of TNF, IL-10, or IL-1, that each cytokine has its own kind of neural signature at the level of the periphery. This is before the signals even go into the brain. Another question we were interested in examining is, can individual neurons in the nodose ganglion respond to multiple different cytokines? So in these, what we call combination experiments, what Tomas did was, you can see these are the spatial ROIs, these are different neurons, and there are some neurons, a class of neurons that respond to IL-1 in an IL-1 fashion, but not TNF. So we call those IL-1 selective. Others that care about TNF, but not IL-1. And then there's another interesting class of neurons that can respond to both. So these are both pro-inflammatory cytokines, but what's interesting is that when this neuron responds to IL-1, it has the IL-1 signature and a TNF signature. There are many other neurons that we didn't focus on in this paper that don't care about cytokines at all. They care about, maybe breathing, changes in pH, mechanosensation, changes in temperature. Those are a vast majority of vagal sensory neurons, but in our Python processing pipeline, we kind of select those out. So this is visualized now as a heat map, essentially. The Y, each row is a neuron over many different experiments. You can see, again, that there's a group of subpopulation of nodose ganglion neurons that respond to IL-1, but not TNF, others that respond to TNF, but not IL-1, and then another group that can respond to both. And then, again, many other neurons that don't care about cytokines. And what we know by doing the immunohistochemistry is that, so if you look here on the right, this is in the supplement of this paper, the vast majority, if not all the neurons, are glutamatergic, so they have the GCAMP6 indicator. And then we have actually the largest proportion that seem to have TNF receptor 1. So this is a cytokine receptor 1 for TNF. This is receptor 1 for IL-1. This is a smaller proportion, and the smallest proportion had IL-10. So presumably, and there are many neurons that you see on the merge, and this is the same is true actually at the nerve level where we're applying the cytokines, but there are many neurons that have all 3 of these cytokine receptors, and then of course there are hundreds of other cytokines and other things we can label with antibodies, but the focus of this paper was on these 3 cytokines. Presumably the neurons that we're responding to in this third class, IL-1 and TNF, also have TNF and IL-1 receptor. We weren't in this study able to match the electrophysiology or the calcium imaging with the exact labeling of the immunohistochemistry, but presumably you need to have the protein, the receptors on those neurons to respond. But we don't know whether or not having those receptors alone will necessarily mean that you'll have a functional response if you record it from that neuron. And then another question we're interested in is, how does now inflammation, in this case colitis, change these neural signals, or does it change neural signals? So we use a model called DSS colitis, dextran sulfate sodium, which you just add to the drinking water of the mice. Mice that are drink that at a certain percentage develop inflammation of the colon. You can score their disease activity. So they get inflammation basically in 7 days. You can see this activity score, and they start losing weight because if you look at their colons, they're shortened. The gut epithelium, you look at the crypts, are become damaged, severely damaged in the DSS colitis mice. Then if you look at cytokine levels, you can see an increase of these three that we're focusing on, IL-1, TNF, IL-10, in addition to other cytokines peaking at 7 and 14 days. But then that resolves in mice that we allow them. So they only get the DSS for 7 days and then it's withdrawn. So there's some mice in these studies where we let them go out to 21 days, and you can see there's a recovery both at the serum level and then at the tissue level in the colon. So then the next thing we looked at was actually how does the neural activity change? Because, my background's as an electrophysiologist and neuroscientist, I wanted to see, how do the neural signals change? Do they change? And if you can see in this video here, again, this is a baseline recording, not with cytokines applied, but basically miniscope on and a control healthy animal versus a sick animal. And you can kind of see, again, qualitatively, that there's actually many more neurons active in the DSS colitis mice. This is at peak inflammation, and you can quantitate that. But another thing that was interesting that we found in this paper was that while there are more neurons active, the amplitude of their calcium fluorescence changes was actually lower compared to control animals. So you can quantitate that again here. So, if we think about that, it means that potentially there's an increase in number of neurons that are active because of inflammation potentially, but every time they have a signal to the brain, it's actually smaller in amplitude. So there's more signals, but they're lower in strength. We don't know exactly what that means. Is that because of cytokines changing in the colon, or is it, it's a kind of a chicken and egg problem? Is it because of the inflammation, or is it related to the inflammation? Is it reflecting the inflammation? Another thing we looked at in this paper, won't spend too much time on this because it's published, is doing RNA-seq from the vagal ganglia extracted from either controls or at peak inflammation. And what we noticed that was interesting was that we had a downregulation of specific genes. This is a volcano plot, so you can see specific genes related to inflammatory signaling were downregulated. This is transcripts, not protein. And then other specific genes that are highlighted here are related to actually membrane potentials and calcium signaling, which is relevant too, because we're doing calcium imaging of neurons and the neural activity. And if you look at the gene pathways, again, there's some interesting pathways that are downregulated related to cytokine signaling and inflammatory signaling, and other ones that are upregulated that are related to signal transduction and action potential generation, membrane excitability. So that now, if we looked at the cytokine-specific responses, again, we saw something very interesting and somewhat unexpected, which is that when you apply the cytokines, like we did in the first set of figures I showed you, in the sick animals with DSS colitis, this is again at day 7 or 8 peak inflammation, we actually saw a change in not all the cytokines, but we saw, so IL-1 did not really change. However, TNF and IL-10 had a significant reduction in the amplitude of the neural responses to TNF and IL-10. We started to think, well, what could that mean? And if we come back to conceptually thinking about what's going on, that when we have inflammation in the body, the signals that we're imaging at the vagal ganglia, the next stop is brainstem structure called the NTS, nucleus tractus solitarius. And we know that then those signals go to other parts of the brain. Some of the connections go to the DMN or DMV, which then is part of this inflammatory reflex circuit. But perhaps what's going on is that in DSS colitis, and of course, this is just one type of inflammation that's induced in a mouse model, preclinical work. Maybe in DSS colitis, what's happening is that because of the inflammation, that the signals that are cytokine-specific signals, if you look here, are becoming degraded because of the inflammation, or maybe because the gut epithelium is becoming damaged. And now the brain is getting kind of this degraded information. So what the NTS sees is then we performed, this is the last figure in the paper, this clustering analysis where we looked at different features of these calcium transients, amplitude, duration, and rise slope. And you can see in the control healthy situation, there's good discrimination between the clusters. because there's a good separation between IL-1 TNF and IL-10. However, in the sick animals, there's much more overlap. So maybe how does that affect the signals now in the brainstem, and then how does the brain deal with that information is the subject of work that's ongoing and future work. But we thought that was pretty interesting. And then on a broader level, again, these signals that we see here, this what we call neural representation of cytokines in these vagal ganglia, these vagal sensory neurons, is that maintained across the peripheral nervous system? Does that hold for other organs? You know, we didn't do pancreas imaging, but there's other work that other labs that have. And so we looked at the colon because, of course, we're inducing inflammation in the colon. We know in mice, and this is also true in the human, the vagus nerve innervates part of the colon. In the mouse, it's the proximal colon. So this is just showing immunohistochemistry of the gut epithelium. You can see Th1 is a nerve fiber label, VGLU2 is glutamatergic, and you can see both in the submucosa and myenteric plexus that you have innervation of nerve fibers, presumably sensory innervation in these areas. That's in the DSS colitis mice likely getting damaged. And how does that change signals? Well, it seems when we do the vagal imaging with the miniscope, in these mice where we apply cytokines now into the colon, not on the nerve, but into the proximal colon of the mouse, we see this maintenance, at least at the level of the proximal colon, of this pattern that IL-10, for reasons we don't understand yet, seems to induce a large calcium transient in these vagal sensory neurons, and IL-1 has a smaller amplitude change. So, you know, work that's ongoing is trying to explore, is there some conserved across the periphery immune neural encoding of cytokine signaling? And the answer is tentatively yes, but you know, more work to be done. So what is the relevance of this, and what's a potential role of the vagus nerve, particularly in diabetes? And a lot of work is happening really in the last 5-10 years. Very exciting work on the sensory vagus nerve, which I kind of that paper that was just published this year is really focusing on the sensory side. And work from actually here at Feinstein has performed vagus nerve recordings. This is all in mice, what I'm going to show you, in animal models. And so what you can do, and what other groups have done, is put a nerve recording cuff on the cervical vagus nerve of a mouse, record, you know, vagus nerve activity in the form of compound action potentials. And you can see there's a lot of activity that happens, and there's work that you can do to try to decode those vagus nerve cuff recordings. And you can see that there are different groups of compound action potentials. This is not the level of individual neurons, because this is just extra neural cuff recording. But there are different groups of neurons that respond to insulin, for example, and there's other work looking at glucose. And so there are definitely glucose-related and signals that travel along the vagus nerve on the sensory side. This is more relevant to perhaps to type 2 diabetes, but work in collaboration with GE Research, with Vicky Cotero and Chris Puleo, using focused ultrasound. So this is a non-invasive modality, just like an ultrasound like you're familiar with going to the doctor for imaging, but focused ultrasound is a different way of stimulation with the transducer to actually stimulate organs and nerves. So in work in collaboration, as I mentioned, with GE Research, they did focused ultrasound targeting the liver, and particularly the portal hepatis, where the vagus nerve innervates. And there are glucose sensing nerves there in that region. This paper is published in 2021, but using focused ultrasound in this work was in diabetic Zucker rat model. They did some work also in pigs and mini pigs. But you can see using focused ultrasound, you can get this change in circulating glucose levels non-invasively. Work from our lab, again, in collaboration with GE on focused ultrasound, again, led by Tomas Huerta, used focused ultrasound, again, targeting the portohepatis of the liver, and showed that you can actually treat in a metabolic disorder, again, more relevant to type 2 diabetes. You can get changes in body weight doing a peripheral focused ultrasound of the liver. And then work maybe more relevant to type 1 diabetes, but very focused on the pancreas from Alejandro Casedo's lab in Florida, showed very interestingly that the nodose ganglion, which I already talked to you about, innervates the pancreas directly, communicating with islet cells there and beta cells that communicate through serotonin. That work also used calcium imaging. Here we can see on this slide, there's definitely tangential and very relevant as far as vagus nerve signaling of metabolism and glucose signals to the brain. There's work, not a lot of work, maybe more work should be done on recordings from the vagus nerve to see, and we could do calcium imaging of the vagal sensory neurons in the context of changes to the liver, changes to glucose levels in the body. And obviously, diabetes, including type 1 diabetes, has nerve involvement. Diabetic neuropathy is one of the very prevalent disorder involving loss of sensory function, particularly beginning in the lower extremities. And that's thought to be the mechanism for that is due to the hyperglycemia, affecting blood vessels. You get a loss of, a blood supply and damage to peripheral nerves, first beginning in the extremities. So there's many different presentations of it, but you could get alterations to the motor fibers that control movement or sensory fibers that give you numbness. So muscle weakness that would be due to motor, numbness would be due to sensory. And then if you look at how the progression of these different neuropathies happen, again, they begin distally. Small fibers are the first one to be affected. And if you look in more severe cases, you might get autonomic involvement, which again, as I just went through, which could include vagus nerve involvement. I don't think that's particularly common, but in a subset of patients that could happen. And there are some symptoms of type 1 diabetes associated with vagus nerve damage, such as gastroparesis, which is slowing of gut motility. We know the vagus, is primary conduit for gut-brain communication and is involved in controlling things in the gut. Of course, the gut has its own enteric nervous system and can operate on its own, but the vagus nerve is definitely involved in sending signals to the brain, and then the brain then reflexively sending signals back down to the gut. Again, these are small studies. There have been cases of vagus nerve atrophy reported in some diabetic patient populations. And we know from work, not so much done in our lab, but in other work, that peripheral nerves, as you can see in this review, innervate the pancreas. Many peripheral nerves do. So it's, I told you about the nodose ganglia and the sensory vagus, which is this light blue line here. There are also sympathetic nerves, and Monica sent me this paper before, but you can see in this study of type 1 diabetes patients that there's a very early loss of sympathetic nerves coming into the pancreas. And, there are some models that for type 1 diabetes, preclinical rodent models that model aspects of this. And you can see, so that would be this line here, but there's also parasympathetic motor innervation of the pancreas. And there's also, I didn't talk about so much, but dorsal root ganglia, which is the somatosensory side, spinal chain signaling, again, sensory going up to the brain. Is there nerve involvement that's relevant to type 1 diabetes? Yes, there are many nerves that go into the pancreas, more relevant to perhaps the paper we just published and other work that we do in the lab. Inflammation and cytokines are involved in type 1 diabetes, and there's increasing awareness that inflammation is potentially central to the pathogenesis of diabetes and its complications. We know that cytokines play crucial roles in orchestrating inflammation, and potentially in the early stage in type 1 diabetes, while the immune system's attacking the beta cells in the pancreas, obviously inflammation could be involved. And now there's, again, not a lot of work, but there's some work showing that inflammatory cytokine IL-1 that I already told you about has been associated with beta cell dysfunction and death. And more of an interesting observation, but alludes to a connection here. Some patients, these are case reports, that are being treated with anti-TNF monoclonal antibodies, they're called biologics, being treated for rheumatoid arthritis, have had spontaneous development of type 1 diabetes. That's one of these papers. And in another case report, destabilization of the existing type 1 diabetes because they're getting treated with a monoclonal anti-TNF drug therapy. So what does that mean? Well, it means that at least for IL-1 and TNF, there seem to be some involvement and interaction with the innate immune system. And that made me think, as I was putting together this talk, what we know about the inflammatory reflexes is it does reduce levels of inflammation in a variety of different contexts. At the end of the talk, I'll tell you a little bit about some work that's very close to becoming clinical for rheumatoid arthritis. But it's based on this discovery, as I mentioned, from Dr. Tracy's lab 25 years ago, published in Nature, showing that if you stimulate electrically at the cervical level in animals, and there's some published work in humans, like I said, in rheumatoid arthritis and some types of Crohn's disease, that you can reduce inflammation, and that's through this cholinergic motor efferent arc. So potentially this pathway that we understand very well, we have a better understanding of that than the sensory pathway, which we're beginning to understand, and hopefully we'll reach this level of maturity. Give us another 10, 20 years, hopefully less. Let's call it 10 years. But we can take advantage potentially of leveraging our reduction in inflammation through stimulation of this cholinergic efferent pathway. And in 2020, we published this paper led by Adam Kressel, who was a surgeon, MD, getting his PhD in lab. And here we use channel rhodopsin animals. So this is an optogenetic study, which is basically optogenetics is a way to put light-sensitive opsins in genetically defined populations. In this case, in cholinergic neurons. So that's why the chat is choline acetyltransferase, which is found in all cholinergic neurons. We put channel rhodopsin in these mice, only in the cholinergic neurons. We know that the DMN, the dorsal motor nucleus of the vagus, is called, sometimes it's called DMV or DMX, is the origin of these neurons that ultimately go down the celiac ganglion complex and the spleen to reduce levels of TNF. So what Adam did here was put a fiber optic cannula invasively to target the DMN. And then when he injected LPS to induce inflammation in the animals that had blue light stimulation, so channel rhodopsin 2 is responsive to blue light. You got a reduction in the TNF, like I showed you already, and that was originally seen in the paper in 2000. However, when Adam did control yellow light stimulation, there was no reduction. And then this paper is published, so I'm not going to spend much time on it, but basically he mapped the neuroanatomical circuit coming from the DMN, going to the celiac ganglion, and then ultimately talking to the spleen. And what we found was that when he injected a retrograde viral tracer HSV1 into the spleen, that traced noradrenergic neurons into the celiac ganglion, which talked to parasympathetic cholinergic neurons from the DMN with another virus that went anterograde into the celiac ganglion. So if you look in that paper, one of the figures shows this convergence of the parasympathetic to a sympathetic population, which again, if you look at the textbooks, there are separate systems that work in a yin-yang fashion and don't talk to one another directly. And this paper was interesting because we show a direct connection between the two circuits. Similarly, in the past 20 years ago, we would say the immune system and nervous system don't talk to each other. Meanwhile, there's conferences every month about neuroimmune interactions. So as we get more data about these circuits and what they do in physiology and how they go wrong in disorders, we can start to rewrite some textbooks, hopefully, and more importantly, you try to understand the neural circuits to treat disease. And so based on this understanding, we have a postdoc working with me in the lab, Kichion Park, who's working on a project to non-invasively activate those DMN neurons using something called Reacher. So Reacher is a red-shifted variant of channel rhodopsin. So instead of responding to blue light, Reacher responds to red light and many other longer wavelength stimulations. And you can see, why does that matter? Well, red light is longer wavelength, so it actually can penetrate deeper through the skin and the skull. And basically, you can test that if you put a laser pointer to your finger, which it's in the lab now. But if you use a blue one, you won't get penetration through your finger. However, you bring in a red one and your finger will light up red. And that's basically the principle we use to activate a population of dorsal motor nucleus neurons that we injected, in this case with a virus, into the DMN, just like where Adam put his fiber optic cannula. But in this case, we're just putting a virus and then activating it non-invasively with red light. So this is actually a fiber optic cannula. The mouse is under isoflurane anesthesia, and it's going into the mouse's ear canal, but not touching anything. And you can see with your eyes that it lights up pretty dramatically the whole head and neck area. And one way we know that we're getting activation of the right neurons, neuronal population, and that the Reacher expression into those cholinergic neurons worked is that we'll see that we have ECGs to monitor heart rate, and we see a reduction of bradycardia, which is the same thing that would happen if you had your vagus nerve implanted with a cuff and you turn the stimulation high enough, you would actually get a reduction in heart rate, because that's a parasympathetic activation, which slows the heart, rest and digest. Then, so that worked. This is work that's not published yet, but work in progress. And when we looked at inflammation, both at the serum level of cytokines, again, looking at TNF, and also in the spleen, we actually, what Kichian saw was a reduction in levels of TNF, similar to what we saw in the other projects. But what's cool here, we think, is that we're non-invasively activating those dorsal motor nucleus cholinergic neurons with the red light. And this is Reacher in the cholinergic neuron. So could a technique or approach like this be used to treat a target that's relevant to type 1 diabetes? Potentially. More work is needed. But what we want to do with these types of approaches is to try these non-invasive optogenetic approaches and others. Optogenetics is one neuroscience modality, but I mentioned very briefly focused ultrasound. There's obviously electrical stimulation, which is the classical neuromodulation that's been around over 100 years. Can we use this type of approach to activate and control neuroimmune circuits? One example of which is the inflammatory reflex in preclinical models of chronic inflammation, such as arthritis, colitis, potentially models that are relevant to diabetes. And ideally we want to do this without anesthesia in a wake animal, and we have work that again is ongoing. This is a mini LED that we can control. We can make this easily wireless, but you can see we can, with this type of light intensity, even with the LED, we're very confident that we can get good penetration of the red light to the deep brain structures if we have a redshifted opsin in the brain. So I'll just wrap up with this study, which is not from our lab, but just came out recently. Monica's probably aware of it because she's very on top of this, but another study that used optogenetics of vagal nerves and specifically to enhance glucose stimulating insulin secretion from beta cells. So they use in this study, again, not from our lab, not from Feinstein, used the channel rhodopsin line that I already mentioned in some of our work, did blue light stimulation at the cervical level to activate that, and reported actually increase in beta cells that produce insulin in the pancreas. What's interesting in this work is that then they also had blue light sensitive microparticles that they put in the pancreatic duct and then activated that. This was actually that's why it's in Nature Biomedical Engineering, and activated with near infrared light, which is pretty cool. And again, if you look in the paper that they saw in mice with the blue light optogenetic stimulation, they actually saw using this marker new beta cells generated. And then they injecting a drug that destroys beta cells. Again, you see this change in blood glucose in mice without the stimulation, but the blue light stimulated animals have a very significant difference. So this is just demonstrating it's possible through optogenetics and through targeting of nerves, in this case the vagus nerve, but it doesn't always have to be the vagus nerve. Obviously, we're biased towards the vagus nerve because we have 30 years of work in it. But this is an approach that we think is interesting and potentially impactful in this emerging field of bioelectronic medicine, which maybe some of you online have heard of. But it's this idea that once you have a molecular readout that you understand, here is the target, and a signal, a neural signal, meaning action potentials, things you can image with calcium imaging, things you can modulate either with electricity, focused ultrasound, optogenetics, something else, but something you can understand the molecular readout, control the signal, and then use a device, whatever that device happens to be. It's important. And that's a new way to treat disease that doesn't depend on pharmacology. The problem with drugs is many times they don't work. Many times they have side effects. In the patients they work when, great, they're likely not going to want to get a vagus nerve implant if they can just take a pill. However, in many patients, the drugs don't work. And in the case of biologics, such as many to treat rheumatoid arthritis, Humira, Enbrel, we see in the US commercials on this all the time. These are anti-TNF monoclonal antibodies. They are immunosuppressant. They have side effects and they don't work in all patients. And also they're very expensive. And so in those patients, this could be an alternative therapy, which is, get an implanted nerve stimulator. This understanding and this approach is very advanced for rheumatoid arthritis. Down the line, there might be this approach for multiple sclerosis or inflammatory bowel disease, which includes Crohn's and ulcerative colitis, which obviously have vagal involvement as well. And what's interesting is also bioelectronic medicine doesn't have to necessarily replace drugs or biologics. And actually in a study run by a company I'll tell you about, in patients that were maintained on biologics, some of them, that didn't get a good response from an anti-TNF monoclonal or a JAK inhibitor following vagus nerve stimulation, they had a different response, a better response. So again, there's an interesting interaction, and what's relevant is that the patients get resolution of their symptoms. In some cases, a bioelectronic intervention, specifically a device or a non-invasive focused ultrasound, let's say, could be used in conjunction with drugs. So again, work from the Tracy lab here at the Feinstein Institutes 25 years ago has now very close to coming to the clinic, and the company is a company called Th1nk Medical, which is actually based in California, in Valencia, California. Late last year, closed phase 3 clinical trials for rheumatoid arthritis. These are patients that failed methotrexate and biologics. 242 patients at 40 different sites in the US. The trial was called Reset RA, and you can kind of see what the device looks like. It's about the size of a fish oil pill, implanted on the left vagus nerve. And this company has now filed for FDA approval. So maybe soon, not yet, but maybe soon in the next months or later this year, potentially, they could get FDA approval for this device. This is from Setpoint. Again, the device, it's a wireless charger, a neurosurgeon, and it's controlled on an iPad. I don't know if it has to be an iPad, but a tablet that the physician controls, not the patient. The device is implanted on the left vagus nerve. It's really an outpatient procedure. A neurosurgeon needs to do it because, as I mentioned before, the vagus nerve travels along the carotid, so you do need a neurosurgeon to do this procedure. But it's very safe. And just in May, so just last month, Northwell Health, and here this is a picture of the building I'm sitting in now. We opened a center for bioelectronic medicine in anticipation that devices like this that Setpoint just closed their trials on. This is a couple news snippets from a patient named Dawn, who's a teacher in Long Island. I think she's a Massapequa, who was a part of this trial that Setpoint ran. She had rheumatoid arthritis, not getting good response through drugs and biologics, and had this implant, and currently has resolution of her symptoms. So, like I said, there's a lot of exciting stuff happening in this space. Northwell Health has opened this Center for Bioelectronic Medicine, which is a clinical center, in anticipation that the patients, once Setpoint gets FDA approval, the first patients to get implanted could get the implant through a neurosurgeon at Northwell and get clinical assessments and treatments and follow-ups at this Center for Bioelectronic Medicine. And, rheumatoid arthritis is probably the most mature, but work from our lab and many other labs around the world in this space to try to understand many different diseases is, I think, going to lead to many more developments of different devices. Not all of them have to target the vagus nerve, but the vagus nerve, as I mentioned, has a good understanding at the molecular and preclinical level of what is going on in the human and how we might intervene in a device-based approach to get symptom resolution. So if anything that I talked to you about over this talk interests you, we at Feinstein are organizing a meeting now in the 7th meeting of this called the Bioelectronic Medicine Summit. It's going to be in New York, somewhere in New York in the fall of 2026. So, mark your calendars now. If you want more info about it, we're just meeting with the scientific advisory board. I actually had a meeting this morning about it. And, everyone who's interested, welcome to come join us. Reach out. I was just talking to Monica before we talked. Monica should come, and we could have sessions that are focused on metabolism and glucose regulation and diabetes. So finally, let me just thank everyone online. Thank you again to Monica for the invitation. I work very closely with Dr. Tracy and other faculty members here at the Feinstein Institutes. Tomas, who did a lot of the work that I showed you, is now at a lab at Choose Lab at Harvard. And these are the rest of our lab, as well as our funding sources. And thank you for your time. Happy to take any questions.
Thank you so much, Eric. This was fantastic. I just want to open it up to questions from the audience.
Yes, great talk. We liked it a lot here. Actually, I'm with the group that was working here with the vagus nerve, both efferent and afferent, and the connection with the pancreas. So,
yes, great work.
Yeah, thank you. My question to you is related to the cytokines. It's the first question related to the cytokines. Do you think that within each organ there is a specialized group of sensory afferent neurons that would sense preferentially the cytokines and inflammation versus other ones? Is there some type of specialization there?
Yes, I mean, this is a question we're very interested in figuring out. We don't know the answer, but what's interesting and also a mystery is that at the level of the vagal ganglia, there doesn't appear to be organotopic, viscerotopic organization. However, when you get into the NTS, there was a Nature paper that they showed there is mapping in the NTS. We are very interested in knowing whether, for example, the sensory innervation of the pancreas that's vagal, how is that organized in some way? Does the brain presumably at the level of the NTS, the brain does know the signals, let's say their cytokine signals are coming from that region. There are experiments that we could try to figure that out, but it's still, unfortunately, we don't know the answer, but we're very interested in trying to do the experiments to answer that question. Of course, in the work we just published this year, there's hundreds of cytokines. Can they all be differentiated at the level of a DFF calcium transit? I don't know. Probably not. So there's likely a combinatorial code of some type. We are, again, very interested in that. So far in what we've looked at with the 3 cytokines, and there's some experiments that didn't make it into that paper, but the signals also change when we combine cytokines. So if we put IL-1 and TNF together, which might be happening in situ, may be happening in the body or in the pancreas, how does the signal change? We don't have a good understanding of that. Obviously, in that paper, we focus on the immune signals and cytokines, but obviously the vagus nerve is also carrying, as your lab's work showed, other signals have nothing to do with cytokines. How does the brain stem first, deal with that information? It almost seems like there's too much information, right? In the mice, we know there's 3,000 to 5,000 neurons at the sensory side mediating this information. In the humans, we're working with more, maybe 100,000 fibers per side, but there's still a lot of information for the amount of neurons and fibers involved. And, that's work. Hopefully, maybe we can work together to try to figure some of this out.
Eric,
Thank you.
We have some really great labs here in the
Yes.
audience. And I've got 2 questions, one from Bart Rope and one from Kevin Otto. Bart, you were first. Do you want to ask your question?
Yes, thank you. Very original presentation, very fascinating. You answered already one of the questions. I was also interested in combining anti and pro-inflammatory cytokines like IL-10 with TNF. What does that do?
So what does that do as far as the neural signals, you mean?
Yes.
I can't tell you. I can tell you offline because it didn't. That wasn't in the paper, but that is a question that came up over the course of when I presented this before. It's unclear. It's not like a 1-to-1, but what we know, which seems to be clear, at least from our data, is that individual neurons can respond. Single neurons can respond to multiple different cytokines. In the figure in the paper, we didn't include IL-10, but we do have the data of IL-10. So it doesn't appear so far, this is unpublished, but it doesn't appear that there are only neurons that respond to IL-10 and other ones that only respond to IL-1, right? There's an idea that in other work, not from our lab, that there are some that dedicated, that carry a signal that only care about this, and they go to a specific part of the brain that only cares about that, and maybe goes to a part of the cortex that only cares about that. So far, at the level of the vagal ganglia, that doesn't appear to be the case. But there are still a lot of open questions. When two neurons are active at the same time that respond to IL-1, and maybe a third that responds to IL-10, what does the brainstem do with that information? We don't know yet, but obviously that's important to try to figure out.
Okay, and if I may, Monica, just
Yeah.
a short question. When the interference intervention studies that you do, I find very fascinating, but how can we avoid that, when you target one organ, you don't mess up homeostasis in other organs that are connected with the vagus?
Yeah, great question. Yes, so one approach would be to target the end organ. So instead of coming at the cervical level, which of course carries all the fibers that ultimately go to, like you said, the lung, liver, pancreas, gut, you could go down to where the organ innervates the end organ. So that would be one approach. There's obviously challenges with that as well, but that would give you a quote unquote cleaner signal as far as the signals that are relevant to that organ and its functions. There are other groups, including groups here at Feinstein, that are developing techniques to stimulate different parts of the cervical level through either current steering, electrode design. There are ways to try to activate different parts. And what's interesting, which is a bigger topic that I won't go into here, is that there's actually very different organization of fascicles across different mammalian species. So In the mouse, it's basically one bundle with a mix of different caliber fibers, myelinated and unmyelinated. In the pig, there's 20 plus fascicles of different mix of different things. In the human, we actually have fewer fascicles than the pig. And in the dog, there's so there's a lot of variability between species. And more confusingly, there's also variability within individuals in that species. So from human to human, there seems to be some difference in the number of fascicles too. We're still trying to figure out how that makes any sense, because presumably all our organs are working and we're getting signals, interoceptive signals to the brain. And in most cases, everything's working great, but there seems to be some variance in the organization of how that happens.
Thank you, Eric. Great work.
Thank you. Thank you for the questions.
And Kevin, would you like to?
Yeah, I'll
Hi,
try
Kevin.
to be fast, Eric. Great talk. I appreciate the platform too. Thank you, Monica. This question is out of left field, and I apologize if maybe you answered it, or if maybe more likely you have no idea. More recently, I've been interested in hypoglycemic awareness and or unawareness. All right. In particular, the recent successful clinical trials were only for patients that were hypo-unaware. They were both type 1 unresponsive, brittle, and hypo-unaware. As a personal type 1 patient, I'm very lucky because I am extremely acutely hypo-aware, and it's I can't imagine being on the other side of this coin. So I'm curious, you talked a lot about the sensors in different areas, especially those glycemic sensors. Is there any overlap, comorbidity with the inflammatory aspect? What makes a person hypo-unaware, and or can we recover it?
Yeah, Kevin, you're right. I don't know the answer to that. However, let me if we could just go back here. So here, presumably the signals involved in these two focused ultrasound papers targeting here were disrupting or altering the signals going up to the brain. Perhaps in a patient that's hypo-unaware, like you said, there's a dysfunction in that signaling, right? And in other chronic inflammatory disorders, such as rheumatoid arthritis, there are reports, and patients report peripheral neuropathies. So there's alterations of different aspects of the peripheral nervous system. Sometimes focus on the extremities, but sometimes dysautonomic, right? So obviously that implicates the vagus nerve. But if you could, coming back to the earlier question, if you could intervene here at the signals and not mess with the signals, involving the other organs, because you're breathing fine and your stomach's working fine, then that would be one way to get an organ specific and maybe function like a glucose specific effect. We're not there yet with, or maybe you have some stealth projects where you're there yet with the device side, but I think that that's a potential possibility in the future, and I don't think that future is too far away. We're making a lot of progress in this space. Obviously, a lot of our work is focused on vagus and immune, but in interacting with a lot of with some patient groups, particularly with inflammatory bowel disease and Crohn's disease, there are a subset of patients, and they might be different from maybe the majority that do have some type of dysautonomia, peripheral neuropathies, and they might have a different type of, for example, IBD than a patient that responds to biologics well, right? Obviously, patients, if you can take Humira or Enbrel or Remicade or something and get resolution, you're not going to want to get a vagus nerve implant, right? And nor are you going to need it. But many other patients, maybe up to half, don't get that resolution. So this becomes bioelectronic medicine. Kevin, you should come. You definitely need to come to the meeting in 2026. I'll reach out to you about it. But obviously, those of us who subscribe to this as a potential approach, this becomes important as an alternative that clinicians, rheumatologists can start to prescribe, which is what we're looking forward to.
And also that technology will likely just continue to improve. So even though it's, sort of whole vagus and invasive with the implantation, potentially in the future, it'll get less and less invasive. I did have one question. I had a bunch of questions, but we're at the end of the hour. I wonder, has your lab explored whether vagus nerve signaling differs between individuals with T1D and those with other autoimmune conditions like RA, lupus, that whole circle of autoimmune conditions?
Yeah, so I mean, there's people, and now you can, on your smartwatch track your heart rate variability, which reflects your vagal tone. I mean, that is a reflection of the heart rate variability, obviously higher being healthier parasympathetic activity is associated with healthier living and lower incidence of chronic inflammatory disorders. That being said, there are groups that have done vagus nerve recording in, I believe, rats and tracked HRV and showed very poor relationship between the two. So there's a very small body of work looking in sepsis and showing that there's increased vagus nerve activity, kind of like what we saw with DSS colitis, right? So there's a little disconnect between we did individual vagal sensory neuron calcium imaging, which is obviously related to vagus nerve cuff recording, but is not the same. So there's some results from vagus nerve recording activity showing that in inflammation of various types in preclinical, this is preclinical, that there's increased activity or alteration of vagus nerve activity. In our work that we just published this year, we show more vagal sensory neurons active, right? We don't know necessarily what that means. And we also didn't do nerve cuff recordings in those mice. So we don't know that would translate to a change in the mice HRV, right? So, I mean, while it's good to track your HRV on your smartwatch, like we don't know that is necessarily the right readout that relates to some of the preclinical work. So that's a roundabout way to answer your question. I don't know exactly about the work for type 1 of diabetic patients and whether or not that's very well understood. What I do know is in different types of autoimmune and chronic inflammatory disorders that there is a subset of patients with like dysautonomia and these alterations in visceral regulation, so that would implicate the vagus nerve. Now, I think last time I was on, we talked about there was one group that did the human vagus nerve recording, but unfortunately, we don't have that human data as far as relating individual neurons to this is what's happening this is how it relates to HRV. Unfortunately, vagal tone in HRV is just a way we can get some indication of parasympathetic activity. And people are very interested in because there's ways you can. I think Andrew Huberman just had a podcast on this yesterday, but you can do humming exercise, breathing exercise. You can buy devices now that stimulate your vagus nerve electrically with ultrasound. You can do these things. So for the most part, the non-invasive approaches are not dangerous. No one's going to in their garage hopefully implant a vagus nerve implant. But these breathing exercises and things that increase your parasympathetic activity are generally good for you, and they're not dangerous. And cold baths, these kind of things do activate the and this has been known for thousands of years. And if there are a way to calm you down and relieve anxiety and stress, great. There's no problem with that. But whether or not they'll be useful, whether or not you can hum or do a breathing exercise to treat a disease is a little bit more of a stretch, I think.
Yeah, that's like a chamomile tea versus a pharmaceutical, I think. But
yeah.
I do. So last question, and we might have to take this to another talk, but do you ever think sort of like just broadly, could the vagus nerve be a target not only for immunomodulation, but also for like regulating metabolic tone? Like if you think about glucagon and somatostatin secretion sort of indirectly, like could there be, off-target benefits, I guess?
Well, yeah. So actually, as you know, but maybe not everyone in the audience knows, vagus nerve implants have been in use and in hundreds of thousands of patients to treat epilepsy and depression, mostly for epilepsy, and then more recently, motor recovery following stroke. So these are brain disorders, but there are a handful of studies showing that in patients getting an implant for seizure suppression for epilepsy, they do have some, like you said, off-target beneficial effects of actually reduced inflammation systemically. The stimulation parameters to treat epilepsy and depression and stroke are very different from what's being used for rheumatoid arthritis. It's actually to treat rheumatoid arthritis, the set point trial, they used 1 minute of stimulation a day and the patients were actually stimulated like at 3 or 4 in the morning while they were asleep. For the vagus nerve implants to treat depression and epilepsy are on in a much more aggressive duty cycle. For epilepsy, they're on basically all day with a on-off cycle to get the seizure suppression. So the biological and molecular mechanisms for these are very different. Obviously, depression, epilepsy, and stroke are brain disorders. Rheumatoid arthritis has neural involvement, but we don't think it's a brain disorder. It might have brain involvement, but the parameters to treat RA and potentially diabetes or IBD would be very different. And that's why it's important to understand the molecular mechanisms first. And this is how bioelectronic medicine might differ from straight neuromodulation or just slapping a TENS unit on something. I could put a TENS unit on my arm and get my hands to twitch. That's not really bioelectronic medicine. So that's why we want to try to understand, obviously through preclinical work, mostly in mice first, but then maybe advancing to rats and pigs before we go into the human, how a circuit is actually working before we put it into a person.
Right. Yeah. Caution. Well, this is super exciting. We keep an eye on the RA progression, indication progression through the FDA, and wishing you luck with all of that. Exciting times for the Northwell new Center for Bioelectronic Medicine. And we look forward to the conference in New York City in the fall of 2026.
Yes.
Thank
Yes.
you again for joining us. It was a great talk. Really exciting.
Thank you so much.
Thank you,
Thank you.
Eric. Thank you,
Okay.
Monica.
Thank you. Thank you for the
Thank you.
questions.
From
Great
Miami.
rest of the day, everyone.
Bye.
Bye-bye.
Take care.